All right, next talk here. This is another nuke special topic. So this time I'm going to talk about mechanisms of localization. So what the hell does that mean? Tracers mimic normal physiology stuff. So, like thallium acts like potassium. Gallium's like an iron analog, like those kinds of things. So, and we've talked about that in prior talks, but what we haven't really focused on is these vocabulary words on how do these things actually get in and out of the cell. So you'll see this come up from time to time. It's not always completely intuitive, so I just want to do a dedicated talk, just sort of run through some of the big mechanisms, and they'll do a few practice questions here at the end. So we'll start out here. So no guessing, show you I'm going to label what the scans are. So this is a technetium systemibi heart. So how does that actually work? How does that stuff actually get into the cell? I'm not talking about how it stays in the cell. I'm talking about how does it get there. So in this particular case, it gets in through diffusion. So passive diffusion, in fact. So I'm going to demonstrate what is passive diffusion. So if we were in third grade and it was science class, we might do an experiment like this. We might take a cup, fill it with water, and then we'll stick a tea bag in it, and then over time you'll see diffusion of the tea into the water. So that's the same thing that occurs in the body. Requires a concentration gradient. It's going to depend on pH and those kinds of things too, depending on where it's going to go, if it's going to cross blood-brain barrier, stuff like that. So passive diffusion for both tech systemibi and tetraphosphate for getting into the heart. And it's also the same way that HMPAO and ECD get into the head, not necessarily how they stay. So like these myocardial agents, for example, they passively diffuse into the cell and then they stick because of electrostatic binding to these negative electric charges that are on the mitochondrial membranes. It's a mitochondrial thing. Maybe like mitochondria. HMPAO gets converted into a hydrophilic species. That's how it stays. ECD gets retained through some enzymatic metabolism stuff that we don't need to talk about. But and that's how they're actually sticking. But how they get in is all passive diffusion. All right. So what about this guy? This is a DTPA study, telling you that, labeling it. So what's the mechanism there? So it's filtration. Now, filtration refers to a special kind of diffusion, which you've got a transit of molecules through pores or a channel. It's driven by a hydrostatic or osmotic gradient, and the primary example of this is glomerular filtration via the kidney. So two primary factors that are actually involved in this filtration. The first one is the size of the molecule. So if they're too big, they're not going to be able to, it's relative to the pore size. So too big relative to the pore size, it's going to get blocked. So for glomerular filtration, only small hydrophilic molecules are going to be able to pass through the pores. The second is availability. So for glomerular filtration, only the molecules that are free in plasma, i.e. not protein bound like the one I just showed, bouncing off there, are going to get pushed through. And if they're small, they're going to make it right through. And like I mentioned also, filtration requires some kind of a force or gradient pressure pushing through here. For glomerular filtration, it's going to be your blood pressure. But it doesn't require local input from any kind of other external energy source. In other words, no transporters, no carriers or receptors. It's non-selective. It's not competitively inhibited by similar molecules, and is therefore not subject to saturation. So filtration, DTPA, that's your GFR thing. So all right, next guy, this guy. So that's a PET scan, right? How do I know it's a PET scan? Well, because it looks like a PET scan, but the other way that I know is that, look at the brain. See how the brain Look at that. That's what a PET scan does. Dark brain, lots of sugar in the brain. So PET works through facilitated diffusion. Now, facilitated diffusion is a different kind of diffusion. It is carrier-mediated. Remember, we said that filtration was not. So we have a carrier. And I like to think about carriers like a revolving door, like at a fancy hotel or something like that. And the reason that I describe it like that is because they can, because you can only sort of fit so many people through there at a time. You can saturate them if you have a lot, like if you have a big crowd of people trying to get through these different doors, like there can be people going through them, so you can saturate it. It's also selective, like you could have a really big fat person, maybe that wouldn't fit through the revolving door, or a little tiny kid wouldn't be strong enough to push the revolving thing, assuming it's not motorized. So there's some selectiveness to the size. Only certain molecules will fit through. It can be saturated. It's still passive. In other words, it still requires a concentration gradient to operate and does not use external energy. So here's an example. I talked about this in the PET one, but like your GLUT channel, and both labeled and not labeled glucose is going to be able to make it through. Things that aren't glucose aren't. There was no ATP involved. There is a competition there, right? That's why blood sugar matters so much. And then like once it's in the cell, then that's a whole different story on how to actually get strapped there with, getting a phosphate group stuck on there, etc. And then like depending on if it's the real deal or not, whether or not it ends up down in the glycolytic pathway. So more on that if you want, you can watch the PET talk. So summary, facilitated diffusion, it's carrier mediated. It's like a revolving door. It doesn't require any energy. You can saturate it. It's selective to the size. You can have, like I said, competition. So summary here real quick. Passive diffusion, the stuff that goes into the heart, stuff that goes into the brain. Filtration, DTPA is the classic one. There is a size limitation, those kinds of things that are dealing with your filtration. And then facilitated diffusion can be saturated. The other ones cannot be saturated, so that's what makes it unique. All right, next one, iodine, 123. So how does stuff end up in the thyroid? How does stuff get into the thyroid? So you may remember this, that the sodium iodine symporter is actually a form of active transport. When I say active transport, what does that mean? That means that it requires ATP, requires energy. It's not free. You got to pay for your metro ticket to get in there. You can't just wander in there for free. Like the other stuff that we talked about before did not require it. And also because it's an active transport system, you can go against the concentration gradient because you're spending money to get in there. So this happens not only with iodine 123 and 131, but also this is how technetium gets in the cell as well. Now you may remember that organification is different between these guys, right? But that's different. That's once it's in the cell already. How does it get in there? Active transport. So some other things also. Thallium, I make a big deal about thallium having to require active transport, living cells, lymphoma, those kinds of things that have ATP. Thallium is a potassium analog. It's a sodium-potassium pump. Rubidium, which is a PET agent for myocardial perfusion scans. It's also an analog of potassium, so it actually falls immediately below potassium on the periodic table, and it also will fit as a potassium analog into the sodium-potassium pump, which is of course an active transport system. So norepinephrine, the uptake receptor there is an active transport thing. It's a symporter with sodium, and it's like the vacuum cleaner. It's bringing the norepinephrine up, putting it in the vesicle, and remember that MIBG is an analog, works on that same thing. So that's also a form of active transport. The cortical scan and DMSA. So remember that DMSA is used for cortical imaging for kidneys, and the way that I remember that is it's looking for scars, and the S is for scar. So, or you could say it's S for sticky, because it sticks to like those sulfhydryl groups, right? So, we've so here's sort of a kidney, and it comes down through into the proximal tubule, and you've got this idea of endocytosis. It's like a fancy word. So it's freely filtered and accumulates in the proximal tubule by endocytosis, which is, you've got these little fibers and things that actually pull it in, create these little vesicles, and it gets in there. And that, again, that requires some energy. It's an active transport system as well. So moving along through there. So that wasn't free. You don't ride the metro for free. So all of these things are active transports. Active A for active, A for ATP. So stuff that goes into the thyroid, including protectinate, that sodium-potassium pump, so any kind of potassium analog, thallium or rubidium. MIBG, remember that is that symporter with sodium, and it's like the vacuum cleaner. Vacuum cleaners need energy to work. Got to plug those things in. And then DMSA also requires, to get stuck, the sticky part requires some energy. So what about MAG3? Remember that MAG3 is secreted, and the way that I remember is that I take that 3 and I like twist it around. So like if you take a 3 and you turn it backwards, look like that. And then I take it and I turn. Anyway, I can like twist that thing in my mind. That works for me, to take a 3 and imagine grabbing the top part with one hand and the bottom part with the other hand and turning it so that it looks like an S. S for secretion, S for MAG3. So what is secretion? Is this secretion? He's pooping. So remember, I collect these defecography cases. Everybody does, right? That's normal, right? It's normal. So basically, secretion refers to a special case of active transport, it's a type of active transport, out of glands or other tissues. So I've got a couple of different common examples here. One is the stomach. You've got your cells in the stomach that secrete hydrochloric acid, but they also secrete protectinate, right? So that's how Meckel scans work. The cells that are capable of secreting this stuff secrete it, both in normal stomach tissue and in the Meckel's diverticulum. The other one is, of course, your MAG3, right? So you've got sort of a diagram of how a kidney works here. So filtration here, secretion here, you actually get some reabsorption here, but I didn't bother to draw that because it doesn't matter. And then this goes into the toilet or into your diaper that you're wearing, your adult diaper. And then the last one is the liver for like secreting HIDA agents like bilirubin, right? But like there's HIDA agents are actually, it's like that secretion of into the bilirubin takes place as well. So secretion, active transport out of a gland and so forth. So tech protectinate, MAG3 secreted, S3 for S, and the HIDA agents. All right, sulfur colloid. So you may remember sulfur colloid works by your reticular endothelial system eating things. So the word phagocytosis is actually a Greek word that means cell eating. So I may not speak French, but I do speak Greek, and everyone who speaks Greek knows that phagocytosis means cell eating. So anyway, I don't actually speak Greek. As if you would think that I would. But yeah, so you've got cells engulfing these different particles. Remember, the size of the particle depends on where it gets eaten. So spleen or the liver or the bones, depending on where it's eating. So, and that colloid particulate gets phagocytosed. So cell eating, reticular endothelial system does the eating. Now, heat-treated RBC scan, Remember, that's done, it's a spleen thing, right? So damaged red blood cells get taken up by the spleen, and there's a word that also starts with S, S for spleen, S for sequester. So the spleen will sequester damaged red blood cells within these sinusoids. When they get sick or they get deformed, it will sort of engulf them and stick them in there, and it won't let them out. And that's what it does. So if you use a heat-treated RBC, the heat is to damage the RBCs. They'll get taken up by the spleen or anything that's acting like a spleen, so like in intrapancreatic splenial splenosis, that kind of stuff. So that's what that's done for. Now, what about the perfusion portion of a VQ scan? What's the vocabulary word for that? So remember, that works by having essentially microembolization or physical trapping. You send a particle in there, and it's just the right size. And because the diameter of the capillary is like 10 microns, you want it a little bit larger than that. So usually, some people will say between 10 and 100. Other people will say between 10 and 50. But you want to get stuck right at the right spot. If it's too small, it'll just pass right through. If it's too big, it'll get stuck further out, and it'll cause a big pulmonary infarct. So capillary blockade is what's actually happening here. So that's the vocabulary word that you want to use if you're asking for a mechanism of localization for the perfusion aspect there. All right, now we've got a bone scan. So how does that work? So it's probably a word you've never heard of before unless you remember it from my prior talk, but it's this chemosorption. So basically, it refers to the binding of a phosphate-type compound onto the surface of a bone. And the strength of that binding is, it depends. It's like in between chemical covalent bonding and hydrogen bonding. So the chemical part is the chemo, and the hydrogen bonding is that adsorption. That's like a physical chemistry term. So it's chem absorption, so it's like in between the middle. So that's where that term comes from. That may not be helpful for actually remembering it, but all right, boom. Okay, so also this isn't just how it works for this, but it's also for how samarium works, and that's one of those old school agents for treating bone pain, like from the 1950s. All right. What about the white blood cell scan? So this is probably pretty obvious, right? So you've got some infection somewhere, and that's like letting off different various signals that your body's like putting off, and then those signals get picked up, smelled like a hound dog, and then the white blood cells come. So it's a cellular migration. The white blood cells migrate to the site of infection, and as a response. So just because you have white blood cells labeled doesn't mean they go to where you want them to go. You want them to go to the infection. Well, how do they go there? It's this idea of chemotaxis of white blood cells that are directed towards the site of infection. So it's a cellular migration, is what you want to say. And then there's this thing. That right there may or may not look familiar to you. This is sort of a newer exam, but it's not too new that can show up on a test. This is what they call a DAT scan, or a isoflupane, iodine 123. So that is normal. It's supposed to look like that. It's a chemical derivative of cocaine as the tracing agent, and it actually binds to these presynaptic dopamine transporters, which is primarily in the caudate nuclei and putamen. So a loss of these dopamine transporters, like i.e. Parkinson's disease, is going to reduce the number of uptake. So instead of looking like a comma, which is normal, it'll look like a circle like that. So that would be abnormal. So normal, abnormal. So another example, same general idea, is this. So what kind of scan is that? Now, that's always the first question. What kind of scan is it? So don't see bones. super duper hot kidneys, right? So that should be a tip off immediately that you're dealing with an octreotide scan. So remember that octreotide works by binding to the somatostatin receptors, of which there are 5 total, but only 2 that bind octreotide. So it's another example of receptor binding. So dopamine receptor stuff, octreotide is binding to somatostatin receptor stuff, so it's this lock and key thing. All right, so let's do some practice questions. I've only got 4 or 5 of them here, but I've tried to pick out some things that might be tricky. So which of the following is not an active transport? So these are obvious that they do use ATP stuff, but then we're down to this one and this one. So I purposely wrote it out like this to trick you. If you do or do not remember that this was actually a symporter also, but instead of calling it a sodium norepinephrine symporter, which would be a giveaway that it was probably active transport, I just wrote it like that. So be careful for sort of tactics like that. So the GLUT transporter, remember that's just your revolving door, so that's not active transport. All right. How does a Meckel scan work? What's the mechanism of localization? So remember, Meckel scans work by pertechnetate localizing to active gastric mucosa. Well, active gastric mucosa does what? Secretes hydrochloric acid. So that's secretion, right? And it's also secreting that tracer. So that is one of our examples of secretion. Sequestration, so remember that the S in sequestration stands for another S, which is an S in the spleen. And I'm going through these quick, guys, but please remember, always when you do multiple choice questions, read the entire question and read all the answer choices. Don't get sloppy. Do that every single time, even when you know the answer, because there may be another answer that's better. Just do it every time. You'll be amazed how many questions you can miss that you know the answer to because you're not disciplined in the way that you take the question. So every time do it the same way when you practice. You cannot afford to miss questions you know the answers to. Okay, capillary blockade. So we talked about this briefly here, and the point being that if it's too small, it goes all the way through. If it's too big, it blocks out early. The diameter of capillary is around 10 microns. These are all in microns. I didn't put units, but they're all in microns. So you need a particle to be a little bit bigger than that, but not too big. So some people say 10 to 100, but 10 to 50 is also, this would be too big. And these would be too small. But that porridge is just right. All right. Last one here. So this is sort of a tricky one. Protectate and sestamib localized. So this is which of the following is true. Localized by the same mechanism. So we're talking about the thyroid here. And so let's sort of rehash that. So remember I said that so go back. So when you do these parathyroid exams, a lot of times there's dual tracer techniques and then there's like dual phase techniques. So that's sort of the general idea for this question. So remember, MIBI is a passive diffusion. Remember we talked about that for going into the heart, and then it gets stuck to the mitochondria once it gets in there. But how it gets in is just this passive diffusion. Versus protectate in the thyroid, it goes in the same way that the iodine goes in, which is through that active that symporter that requires active ATP. All right, so that one's not right. So thyroid uptake of sestamib is not affected by TSH. So TSH actually stimulates that sodium iodine symporter. So if somebody was taking Synthroid, that might suppress the TSH and decrease the uptake of everything that uses that symporter, which is the 123, the 131, and the protecnetate. So, but MIB goes in through diffusion, right? And that is not, you can't saturate that. So it doesn't. It's not affected by TSH. It's going to wash in regardless. Now, so this is sort of. So this is true, but it's not localized through active transport, right? That's the 123, 131, and protecnetate. So that's wrong. And then. This is sort of not true either, right? So that's the exact opposite of the mechanism for how that works. All right, so that's it. That's the end of this talk.